Hi everybody and welcome back to this short course of bioinorganic chemistry. We have discussed so far the importance of inorganic elements in biology and also have showcased how our life affected directly out of such elements. I also have highlighted in my last 4 lectures the beautiful design principles adopted by nature in controlling some of the key chemical processes that are extremely important for our life. Now, we will talk about life with oxygen. Let us go back to see that molecular orbital diagram of dioxygen. You are all familiar with that. So, this is sigma 1 s, then sigma 1 s star, sigma 2 s, sigma 2 s star, and then sigma 2 p z and pi 2 p x 2 p y, and the antibonding orbitals is having 2 unpaired electron in the pi star 2 p x and pi star 2 p y. So, 2 unpaired electrons, so we call it as a triplet state. So, molecular dioxygen is in triplet state. As you can see that antibonding orbital 2 unpaired electron, and that is why we call it is triplet state. Now, what would happen if you oxidize by 1 electron, then the electron which would be removed is from antibonding, then it will form dioxygenyl ion O2 plus, and if we reduce it by 1 electron, then 2 electron, it will generate O2 minus, which is called superoxide ion. In the superoxide ion, as you can see that there is only 1 unpaired electron now, and after 2 electron reduction it is O2 2 minus, it is called peroxide ion. In the peroxide ion, as you can see that 2 p x 2, 2 p y 2, both the pi star antibonding orbitals are filled, and this is what I compare between O2 plus, dioxygen, O2 minus, superoxide and peroxide. If you look at this bond order in O2 plus, as you can see it is 2.5, O2 it is 2.0, O2 minus it is 1.5 and it is 1.0. So, bond order is decreasing from left to right. And what happens to the distance? Bond distance is gradually increasing from dioxygenyl ion O2 plus to O2, then O2 minus, then O2 2 minus. Bond dissociation energy, as you can see, if O2 plus, certainly the bond dissociation energy would be very large, 148.8 kcal per mole, and then O2 117.0, O2 minus 88.0, and O2 2 minus it is remarkably less, it is just 46.0. So, you see that peroxides having a very small bond dissociation energy, and stretching frequency will decreases from O2 plus to O2, O2 minus and O2 2 minus. We all talk about reduction, but these oxidations of dioxygen, as you can see, has been achieved by Neil Bartlett long back, and what he did, he took this O2 gaseous oxygen, PTF6 also another gas, and once they combine it forms red solid O2 PTF6, and where actually O2 convert to O2 plus. bond error increases, it is dioxane oxidation. And this has been very clear. Presence of O2 plus with the STO stretching frequency is 1838 centimeter inverse, which confirms that it is O2 plus. That distance is 1.13 angstrom. So, all these characteristic features confirms the presence of O2 plus in this molecule. Once this Professor Bartlett looked at this molecule and he saw the first ionization potential between oxygen and xenon. In oxygen it is 12.2 electron volt and in xenon it is 12.3. Then he was surprised that oxygen and xenon, as you all know, xenon is a inert gas and inert gas will not react at all, and that is the reason why it has been kept separately in the periodic table. Having seen this first ionization potential, he could make this O2 PTF6 successfully isolated as a red solid. It inspired him to look for the synthesis of very similar molecule XCPTF6, which he has achieved, and as you can see that the xenon gas and PTF6 gas forms a beautiful orange yellow solid, and this was first noble gas molecule has been synthesized in the laboratory. So, this tells very clearly that noble gas, which is supposed to be highly inert, can also be reactive, and this is the beginning of a new era where the noble gas molecules started synthesizing in laboratory across the globe. In the textbook you will find so many noble gas compounds. However, this is the first molecule which was synthesized in the laboratory by Neil Bartlett. So, it is a remarkable achievement. Let us look at the dioxane reduction, which is very popular. So, dioxygen O2, after one electron reduction, it will form O2 minus, as you have just seen, it is called superoxides. Further reduction gives rise to O2 2 minus, with the proton it forms hydrogen peroxides, which are very popular chemicals in the laboratory. If you reduce one more electron in presence of proton, you get water and OH radical and hydroxyl radical. If you reduce one electron and have one proton together, it also forms water molecule. So, this is the dioxane reduction, and we will now look at very carefully their redox chemistry. This dioxygen, as you can see, O2 to O2 minus, the potential is minus 0.33 volt, and then O2 minus to H2O2 plus 0.89, H2O2 to H2OH radical is plus 0.38, and then 0.89 to H2O. However, if O2 you convert to water directly, the potential that is required is plus 0.815 volt. This has been tabulated over here. This is the first reaction, one electron reduction O2 to O2 minus, which produce a negative potential of minus 0.33, and indeed all other potentials are very positive. The most important reaction, that O2 plus 4H plus 4 electron gives rise to 2 water molecule, the potential is 0.815. The reduction potential for the 4 electron reduction of dioxygen actually shows the great oxidizing power of dioxygen. This reaction indeed represents the major source of energy in aerobic organisms when coupled with oxidation of electron rich foodstuff such as glucose. Let us look that one electron reduced species O2 plus electron O2 minus, as I have shown that the potential E0 is negative minus 0.33. Now, if you convert to Gibbs free energy delta G, you will see a positive values. That means, this reaction is not a favorable reaction. However, if you look at all other reactions, E0 is all high positive, highly favorable. That means, the oxidizing power of dioxygen, which is clearly reflected at the end like O2 plus 4 H plus 4 electron gives rise to water with a potential of 0.815. This is highly favorable, but this one electron reduction is a unfavorable reactions. So, one electron reduction of oxygen to O2 minus presents a barrier that protects vulnerable species from the full oxidizing power of dioxide. However, to get the full oxidizing power, this 4 proton and 4 electron have to be given in a concerted way, so that it produce water and this oxidizing power of dioxygen can be utilized. If you look at the hydroxylation and epoxidation reactions, some of these reactions I am showing, like methane gas plus half O2, which is all in gaseous phase, it forms methanol and delta H is minus 30 kcal per mole. It is actually an exothermic reactions. Benzene converted to phenol, all these reaction of dioxygens are indeed thermodynamically favorable and highly exothermic, but generally very slow in absence of a catalyst. So, although it is a favorable reactions, it never happens just by adding this, you need a catalyst to get that product. Say for example, hydrogen and oxygen is giving rise to water, it is a highly favorable reactions, but it never happens in environment, otherwise we cannot survive, because all hydrogens and oxygen which are present in environment will combine and gives rise to water, and then this universe will not survive, but it never happens. What is the problem? The only thing standing between you and spontaneous human combustion is the triplet nature of dioxygen. Say organic molecules are all spin paired and thus it is called singlet. However, a dioxygen which I have shown you, there is 2 unpaired electron and in the ground state of course, and this is triplet. Thus, the singlet and triplet, they are actually spin forbidden. They generally do not react and the reaction would be very slow. So, this is what is the problem. Okay. So, this is a kinetic barrier that the triplet oxygen, if you give 22.5 kcal per mole energy, then triplet convert to singlet and then there is no problem. The singlet oxygen can react any organic molecule and it is a spin allowed. However, there is a barrier, we call it as a kinetic barrier. They have to overcome this energy to reach to the singlet and then this oxygen is highly reactive. You can see yourself in the respiration what is happening, the glucose which is, as you all know, organic molecule, the singlet and The normal atmospheric oxygen, which is a triplet oxygen, will not produce 6 CO2 plus 6 H2O plus 671 kcal per mole. So, this pathway is a forbidden pathway. The reason is that it is spin forbidden. So, one is singlet, dioxin is triplet. So, it will not happen. When it happen? When this dioxygen is activated by giving an energy of 22.5 kcal per mole, then this triplet dioxin convert to singlet dioxin, which immediately reacts with the glucose, gives rise to carbon dioxide, water and huge amount of energy. This is what is happening in respiration. So, dioxygen need to be activated in order to react with the glucose, which gives rise to lots of energy, which is the source of most of the energy we have in our day to day activity. Let us look at this electronic states of molecular oxygen, as this is the ground state, which is sigma, is a triplet state, this is 1 by Z, and the first excited state is a delta state, it is singlet 1 delta Z, and the energy which is required to convert from a triplet to singlet 1 delta Z is 22.5 kcal per mole, and if you add like 37.5 kcal per mole energy, then it will lead to another singlet state, the next excited state. You see that this is also singlet, but one is up spin, one is down spin. So, eventually it leads to singlet state, and 1 delta Z, you see that both the spins are paired and leading to a singlet state. Of course, you need to provide a energy of 22.5 kcal per mole to reach to that state. Then the dioxygen, which is otherwise not a reactive species, becomes highly reactive. It reacts immediately. Organic substrate, we will see some of this reaction. For example, it is a highly reactive singlet oxygen. Once we produce this singlet oxygen, it immediately react with the organic substrate, and there is a large number of substrate which is there in any textbook, you can find it. I am showing just 3 examples to let you know that how reactive it is. Like this singlet oxygen, it produces like O bond, as you can see over here. This is in type reaction, and if you have a double bond with the singlet oxygen, it can form a cycloaddition 2 plus 2, and if you have something like this with the singlet oxygen, it produces a cycloaddition 4 plus 2. There are many examples known now where the singlet oxygen shows remarkable reactivity. This singlet oxygen, as I have already said, that you require something 22.5 kcal per mole, which is a huge amount of energy. You all know the Jabrowski diagram very well. What is exactly happening if you have a photosensitizer, say it is a ground state S0, and you shine light, and then what would happen? The photosensitizer will be in excited state. Of course, this is also a singlet state, and there are many excited states like S1, S2, S3, S4, and after non-radiative transmission, it can comes to this S1 state, and this can go back via the internal conversion to the ground state again. And if it goes back to the S0 state, this will produce is called fluorescence. However, via intersystem crossing, it can go to the triplet state, and then from triplet it goes back to the singlet state, the ground state, and you see that this is not a favorable transformation triplet to singlet, and this would be a slow, and this is phosphorescence. This triplet state is a high energy state, as you can see. These can transfer energy, and dioxygen, which is the triplet state, can take that energy and convert to the singlet oxygen. So, coupling of these two leads to the formation of this singlet oxygen, and this singlet oxygen can be utilized for various chemical transformations. I will show you some very interesting applications which all of you are aware of. Now, this singlet oxygen also detected spectroscopy. You see that tetraphenyl porphyrin, just like I have shown that if we irradiate triplet oxygen with light in presence of tetraphenyl porphyrin at minus 30 degree centigrade for 3 hours, then it will give the singlet oxygen, which will react with 1,4-dimethyl naphthalene and produce these types of endoperoxide intermediate with T half around 5 hours at 25 degree centigrade. Converts back to the parent molecule and releases very reactive singlet oxygen, which, as I have shown, it can be detected in phosphorescence. This, the peaks comes at 1270 nanometer. When it is coming back from delta to the sigma to the ground state, the triplet state, it produces phosphorescence band at 1270 nanometer, which confirms the formation of singlet oxygen during this process. The singlet oxygen is highly reactive, and it can be utilized by it for various chemical transformations, various important applications. So, one of these very important applications is the PDT drug, photodynamic therapy drug. For example, what is happening that the substrate, if you give photon the light, what will happen? The photosensitizer will go to the excited state, H star, and as I have shown earlier. So, this state can transfer the energy to convert the triplet dioxygen to a singlet dioxygen. It is a coupled process. And once this you form a singlet oxygen, the singlet oxygen, as I have said, that highly reactive. Indeed, the cancer cell, if you give the singlet oxygen will burn this cancer cell, and the cancer cell becomes dead cell. This is one of the very popular technique that are being used as a photodynamic therapy. And photoprin, which is actually a oligomeric mixture of this multiporphyrin molecule, and this is a photosensitizer, that is what is being approved so far and popularly utilized. Now, there are many PDT drug known today which are showing better activity, and this is very promising field of research. You see that what you are using? You are using light and dioxygen. Both are very friendly, and as a result of that, and of course, you are using a photosensitizer. Here it is photoprin, but you generate a singlet oxygen which is highly reactive, which are used to kill the cancer cell. Okay. These techniques, here I will give little bit more information about this photodynamic therapy. Photodynamic therapy is relatively new treatment in cancer therapy, first used in 1978. there is currently one PDT drug, photoprin. This drug is currently approved for the treatment of cancer. So, what is actually happening in this PDT, photodynamic therapy, that if you shine light, so most useful light is around 630 nanometer, which is called red light. Why red light? Because red light penetrates the cell more than the other light. So, that is why red light is so important. So, red light actually penetrates the tissue and it reaches the point. So, how it is happening? For example, here this is a diagram is showing like PDT with photoprin, like the patient having tumor cell. Say for example, here it is tumor is there, is shown over here is a tumor. So, 40 to 50 hours before you are injected the photoprin, the photosensitizer is basically 2 milligram per kg, and slowly you are added this photoprin, and then this photoprin will selectively, of course, it goes in other parts of your body as well, but it also concentrated to this tumorous cell. And then if you shine a light, a red light, and produce singlet oxygen, then cancer cell will be burnt out of those singlet oxygen, then your cancer would be cured. So, this is such a simple technique, cost is much less, and you are only using dioxygen and light for this process and produce singlet oxygen. So, these are the various applications that one can utilize this singlet oxygen. Now, coming back to this triplet oxygen and singlet oxygen. So, there is, as I have said, that triplet oxygen, if you give energy of like 22.5 kcal per mole, you produce singlet oxygen. So, this is a kinetic barrier one has to cross, so that this singlet oxygen can then be highly reactive, and you can do that in the laboratory. This 22.5 kcal per mole energy is too much for a biological system. Now, how do enzyme overcome this kinetic barrier? Now, this is what is called dioxin activation. Indeed, the dioxin need to be activated. This amount is too much for the biology. One need to reduce this energy in order to make use of this singlet oxygen. Otherwise, it is impossible to do that. Okay. So, dioxin activation, these days we are hearing this. This is basically how biology can reduce this energy, so that it can be affordable then. Okay. So, I will give just two examples to show you, just tell you that how biology actually bypass or overcome this kinetic barrier. Like I will be discussing in details, indeed, the cytochrome C oxidase, which actually produce energy, where dioxin gives rise to water and release huge amount of energy. So, what is happening that if you add 4 proton 4 electron in a concerted way, this is thermodynamically favorable, then you do not go through that 1 electron reduced species. So, dioxygen binding site contains 2 paramagnetic metal centers, one is iron, one is copper, where dioxin binds and it actually forms O2 2 minus. Dioxin is in triplet state, is a paramagnetic. So, the paramagnetic 2 paramagnetic metal center binds the dioxygen, and then it reduces by 2 electron to produce peroxides, and thereby bypassing this unfavorable 1 electron reduction process to form superoxides. Now, I will give another example which we will discuss in details in subsequent lectures, is that cytochrome P450, another very popular enzyme. This enzyme catalyzes the reaction of dioxin with organic substrates. Here also the dioxin binds at the paramagnetic metal ion, okay, and thus overcome this spin restriction. So, your dioxin is triplet paramagnetic, your metal is also paramagnetic. So, no problem. So, it overcomes this kinetic barrier and reaction is possible, and nature utilize this path to overcome this kinetic barrier. There are many such examples, not all enzymes go through the same way. Some enzymes activation is required. I am not going to discuss today in details. In summary, I have discussed today how we are protected from the great oxidizing power of dioxygen. We talk about thermodynamic and kinetic barriers for the reactions with dioxygen, and also how one can overcome that. In my next lecture, I will talk about how dioxin is utilized in respiration to produce huge amount of energy that we use every day. Thank you.